My name is Ellie Hoover, and my topic is Urbanization is harmful for digestion in honeybees. Let's get into it. Honeybees, also known as Apis mellifera, have been facing decline in their populations for several decades. Honeybees are very valuable to us and they provide pollination services, which are estimated to be at least $235 billion annually. One of the many threats causing population decline is urbanization. Urbanization leads to pollution, which impacts honeybee digestion of their main source of food, which is pollen. A little bit of background regarding honeybee digestion. The organ that we're going to focus on today is the midgut, which is responsible for nutrient absorption. Important structures are the villi and the brush border that maximize the surface area for maximum nutrient absorption. In the diagram at the bottom, you can see the purple structure with the protruding structures. Those are the villi, and surrounding them are the brush border. Within the brush border, there is an enzyme called aminopeptidase N, which is responsible for the digestion of pollen. Urban pollutants are anything that are environmentally toxic that have human origins. The ones that we're going to focus on in this study are mercury, lead monoxide, and cadmium oxide. Another thing to consider here is the urban heat island effect. This is essentially a heat inversion that's observed in cities. Cities tend to be hotter than the surrounding areas because concrete holds onto heat a lot longer than vegetation. My research question is, how does urbanization impact digestion in Apis mellifera? Why is this important? Honeybees rely on pollen as their main source of food. If urban pollution is present in the pollen, honeybees absorb the pollutants into their digestive systems, which causes harm and makes it harder for them to digest pollen in the long run. The first case study we'll focus on covers the topic of the detoxification gene expression in honeybees. Jazza and others collected both honey and honeybees from 3 different regions: urban, agricultural, and mountainous. They tested the midguts of nurse bees for mercury and lead levels and the expression of the following genes that are associated with detoxification. In both studies, they found that honey and honeybees from urban environments had the highest levels of mercury and lead and the highest expression of detoxification genes. The next case study focuses on the topic of cellular changes that are observed in the midgut. Tabor and others collected honeybees from an experimental apiary. They were fed sublethal concentrations of cadmium oxide and lead monoxide for 9 days. Transmission electron microscopy, or TEM, was used to view the midgut cells of honeybee workers. In groups A and B, as pictured, that was the control group, and you can see that it just has typical morphology in the cell. Groups C and D were treated with cadmium oxide. They were observed to have lysis of the nucleus, which just means rupturing of the membrane, cytoplasmic proteolysis, and elongation of the mitochondria. Groups E and F were treated with lead monoxide, and they were observed to have swollen mitochondria with fractured cristae and swollen lysosomes. Groups G and H were treated with a mixture of cadmium oxide and lead monoxide, and they were observed to have swollen lysosomes and enlarged phagosomes. The last case study we'll focus on covers the topic of aminopeptidase N activity. Ferrari and others selected 10 sites in Milan, Italy, that were various locations on the land use gradient. This is just the gradient from rural to urban. They extracted the midgut of bees and measured the APN activity, aminopeptidase N. The APN activity generally decreased with warmer and urban sites. They used temperature as the main factor for determining urbanization due to the urban heat island effect. To conclude, with our first topic, detoxification gene expression. Honeybees from urban environments had the highest levels of detoxification gene expression. The second topic, cellular changes in the midgut. Honeybees exposed to urban pollutants experienced various cellular component lysis, proteolysis, and swelling. The last topic, aminopeptidase N activity. Honeybees with warm urban environments had the lowest levels of APN activity. In conclusion, urban pollutants and the heat island effect negatively impact honeybee digestion. Something to consider, is urbanization all bad? One study that I researched found that urban cities provide more diverse foraging areas than meadows. They also found that bees are more sensitive to agricultural pesticides than other pollinators. Lastly, they found that honeybee flower visitation is higher in urban areas than in rural areas. While all of this is true, and it is important that urbanization allows access to more diverse foraging, you can't really compare agricultural pesticides to urban pollutants because they're both harmful. Lastly, the significance. The long-term impact of these findings implicate that honeybees will continue to have population decline if their digestion is impacted. For honeybee biologists, we need more research on specifically how changes to the midgut observed by the urban pollutants affect honeybee health. We learned about lysis, proteolysis, and swelling of these organelles, but how exactly do they affect the honeybee health overall? For honeybee management, navigating Apis mellifera management in urban environments with this knowledge means minimizing exposure to the pollutants causing their digestive impairment. As a city resident, what can I do? You can advocate for proper pollutant management from your city council and volunteer for cleanups. And these are the references. Thank you.